Welcome to the lesson on transcription. It's always important to keep the big picture in mind, and the big picture here is how do we get from DNA to actual physical traits, like for example, eye color or height or hair texture. And it begins with the DNA, and the DNA is organized in these chromosomes. Eukaryotes have multiple chromosomes. Humans have 46 of them. And those chromosomes are made up of very long chains of DNA. And the DNA has segments on it called genes. And genes are pieces of DNA that actually code for a protein. Before that gene can actually be expressed as a gene, as a trait, several things have to happen, namely transcription and translation. So first, we're going to take one of the strands of DNA and make a kind of copy. And that copy is here. It's called messenger RNA, which contains the message that's inside of the gene. And this copy can be taken out of the nucleus because the DNA has to stay there. It's our master copy. But the mRNA can be taken out of the nucleus, and it's going to go to the ribosome, which is our protein factory. And in the ribosome, we make the protein. We decode the message that's in messenger RNA, and that message gets changed into a protein. And it's proteins that allow you to express your traits and make you the way you are. Like there's proteins that give you the different eye colors that you have. All right, so last time we talked about RNA, and we talked about 3 different types of RNA, so this is a quick review of that. We have transfer RNA, which is going to bring in the amino acids so we can build a polypeptide, which is going to make our protein. We have the ribosome itself, which is made up of RNA. It's made up of rRNA, or ribosomal RNA. But today's lesson, we're going to focus on the messenger RNA, mRNA, and how that is made. It's made through the process of transcription. All right, so protein synthesis, like we discussed, involves transcription and translation. Today we're going to focus on transcription, and in the next lesson we'll look at translation. So transcription involves taking one DNA strand inside of your gene and using it to make a kind of copy, and that copy is called messenger RNA. So that's transcription. And in the next lesson we'll look at how that information, that message, is decoded and turned into an amino acid sequence or protein. So how does transcription actually happen? So during transcription, the first thing you have to know is you're only going to take a little piece of the DNA and make a copy. So in DNA replication, which is different, in DNA replication, we were copying the entire DNA strand because we wanted to make daughter cells that each had their own copy. In transcription, we're not doing that. We're just making a little copy of a segment of the DNA that we're interested in currently, and that segment is the gene. So for this reason, RNA is much, much shorter than DNA is. Now, the enzyme involved in this process is called RNA polymerase, represented by this green oval. And what the RNA polymerase does is it's going to bind to the promoter sequence. And the promoter sequence shows the RNA polymerase where transcription has to begin. And that promoter sequence is very close to the gene that you're trying to transcribe. So that's the first thing that happens. The second thing that happens is the RNA polymerase is going to unwind the DNA. So you can see here the DNA is unwound and separated. And it's going to start adding in new nucleotides, RNA nucleotides. And here they are. The brown is your RNA strand, and you can see it's being added one nucleotide at a time. And if you remember, the RNA nucleotides are A, C, G, and U. There's no T in RNA, and that's what's being built over here. The other thing that you should notice is that only one side of the DNA is being used. The other side is not being used because we only need to make one strand. RNA is single-stranded. All right, so we follow normal base pairing rules. So C goes with G. A would normally go with T, but since we're making RNA, it goes with U. And it just continues using base pairing rules. And if you look at the brown strand, the RNA that's being made, it's almost an exact copy of this strand of DNA over here. So we have a G, we have a G down here. We have a U here, and we have, what the equivalent of that is in DNA, which is T. We have a G over here, we have a G over here. So that's what we mean by we're making a copy of the DNA. It's not an exact copy because we have uracils instead. Okay, so that is transcription in a nutshell. We end up making this long piece called RNA at the end of it. Now, it's actually not finished. So transcription at this point is not complete. We have to do some editing to the RNA. So if you look at RNA and if you look at DNA, it's got these pieces called introns. And that these introns are basically non-functional segments of DNA, and we need to get rid of them. The exons, on the other hand, are the pieces we're interested in because these code for a specific protein. So we have to kind of remove the part that's almost like junk. So we want to remove these introns, we want to cut them out, and we want to glue the exons together. So this is exactly what happens during mRNA processing. We cut out the introns and we glue the exons together, and we glue them together using an enzyme called ligase. And at the end, you have the exons only and the introns are gone. The final thing that has to happen is we have to add a cap and a tail to finish your mRNA piece. And the cap is actually something called guanine triphosphate. It's added to the 5' end. And this is going to later help this mRNA bind to the ribosome when we get to translation. And the tail is a poly-A tail. That just means there's 50 to 300 adenines. So 50 to 300 A's added to the end of the mRNA strand. And that kind of sends the message that this mRNA is ready and it's done and it's ready to go and leave the nucleus and find a ribosome. So at this point, your mRNA is complete. So back to the image we started with. We just built an mRNA, a messenger RNA. It has that secret message that's inside of the DNA, that's inside of the gene, that's going to tell us how to make a specific protein. And maybe this specific protein is going to be melanin, and it's going to help make a person's eye color unique and the way that person has their eyes. Right? So this is exactly what we made over here. Next time we're going to look at how this message is decoded. How do we make sense of the information that's here in order to make proteins? So thank you so much for listening.